Caballeros y damas, buenos días. It's great to be in Madrid here and having the opportunity to meet with you. And today I would like to touch on two relatively recent pieces of work that we did in Innsbruck. And for that reason, I tell you, I don't know about you, but I do like newspapers very much. Because the newspapers, they tell me on which cases we are working on. They usually learn about that case much earlier than we do in our laboratory, like it was in the case with the identification of the Mexican students. Now with that case, and that, as you see later, links to massively parallel sequencing, we were asked to identify 17 pieces of evidence which came by carriers from Mexico. And those specimens were in a really bad shape. If you get such specimen in your laboratory, you close the box and send it back. This is completely burned material, exposed to water, and it's small in size. So, you even don't want to start to work with it. We promised to try, and we were trying to extract DNA. And following the New York Times, we were lucky because we were able to get a full STR profile out of one of these specimens. To me, that was almost like a wonder because the samples really looked very bad. So we got, using two different STR kits and PCR-CE-based approach, we were able to get the profile that matched the family references. That is the second wonder, because it's unfortunately practice in Mexico that people die this way, that we are so lucky to have a piece that works and gives a DNA profile and then matches one of these 43 families that are searching for their relatives was really a big piece of luck. On the other hand, as BBC rightly points out, we were not able to get anything else for the remaining samples. The quantities that we got by real-time PCR, and we are using multi-copy targets for both mitochondria and nuclear, were 0. So there is no difference from background. And you know what's going on? Very likely, the tissue is burned, the DNA is burned, but we ask ourselves the question, is there a possibility to find at least some DNA that is highly degraded? And that reminds ourselves about the experiments that you do in your laboratories, and we do them as well, trying to get an understanding of the DNA quantity by looking at a certain DNA quality. In our routine work, we are using a target for mitochondrial DNA quantitation that is 143 base pair in size. That means if we get a quantity, it relates to DNA fragments that are in that size range or larger. Now, when we move down in the size of that fragment to 57 and 51 base pair, that's the red line and the blue line that you see in the graph here, and expose DNA to degradation by DNase treatment, we do see that the 143 base pair fragment is linearly decreasing in quantity, while the smaller fragments are still detectable at a higher concentration, which is what we would expect from DNA that is going to get degraded. If we look at the same plot in a logarithmic function, we do see how that story ends in the longer exposure times, and we see that reaching a level of about 1,000 copy numbers mitochondrial DNA, which in some scientific disciplines is considered a reliable threshold above which we are getting good mitochondrial DNA results, we are reaching these 1,000 copies with the 143 base pair fragment after 8 minutes under these conditions, while it takes more than half an hour for the very short ones. So this principle is all familiar to us, and if we try to demonstrate that by experiment, it's working. Of course, this is artificially degraded DNA, Now we are getting an interest, how would that look like with DNA that is undergoing natural degradation? And in that case, we were trying the same experiment using bone samples, DNA extracted from bone samples. They come from a medieval grave. It's more than 1,000 years old. They went through substantial degradation over time. And when we do the same experiment, we clearly see, I'm picking two samples here, a blue one and a red one. With the 143 base pair fragment, we do see very little copy numbers. Note that this slide is showing the y-scale in a logarithmic function. So we have about less than 100 copies for the red sample and nothing to detect for the blue sample, which is more or less the same in this range of quantitation. But if we look at the shorter fragments, we are getting 20,000 copies. So even those samples, and this is also what archaeogenetics is telling us, are holding useful DNA information if we restrict the size of the amplicon or the fragment that we are looking at. Of course, such small samples cannot be, or such small amplicons, cannot be targeted very well with the conventional PCR approaches that we are using, right? Because we need to fit two primers, and then we want to see a region between the primers that is giving us information. So if we would use like 2 times 20, I don't know, 20 base pair primers or 25 base pair primers, we are using up the 50 base pairs already without having any substantial information about a target inside. And this is why I thought, well, we should explore more the area of massively parallel sequencing, because there we don't have this limitation. And I was talking to two wonderful PhD students that I have in my group, Katrina and Myra. They are both working with such markers and with such technology. And I wanted to make them enthusiastic about, going in that research and trying to optimize what we are doing. And I asked them to come to my office and have a coffee. And as you see, they were not really picking this up from the first moment. However, later they got more and more motivated doing that kind of work. And we were pursuing this. So they were screening the literature for alternative library building approaches than what we are using for conventional PCR systems. And there we learn a lot about the papers and the studies that have been done on very old specimens, like the Neanderthals. And the method that was striking us is a method that is referred to the PEC method, which is primary extension capture. And the principle is pretty straightforward, as in most of these cases. A target specific sequence is used to capture DNA from the extract. First of all, the DNA is linked with adapters. In that case, it's a 454 sequencer. So they have the A and B adapters. We modified that to fit in the PGM pipeline. So we were using the A and the P1 adapters. And after ligation, those target-specific primers bind, and you would do one single round of PCR, or if you want, one elongation, to increase the stability of this complex between the PEC primer and the target. And if that is successful, you can get out this construct, wash away the rest, do the enrichment, load it onto the beads, put it on the chip, and taking advantage of this pipeline. We tried it with the control region first, because that's a region that we are very familiar with. And you see here an alignment of this first sequence to the reference sequence. The control region spans from a position that is referred to 16024 to 16569, so it's the very end of the molecule, and from position 1 to position 576. Our primers were designed in a way that we can use a little more of just the control region. And you see we have nice coverages. The red graph corresponds to forward sequences. The blue graph corresponds to the reverse sequences. What you also see is there is some signal in the middle of the molecule. We were using the primers that we use for mitochondrial DNA quantitation, also in this PEC assay. And of course, we are getting a sequence that is related to this region around position 8,300. So that tells us, depending on which primers we are putting in this reaction, we will get target-specific sequence if there is some. Okay, so what do the results mean? We were looking at those old medieval samples because these are the samples that we have a pretty good understanding. We have been working with those on different occasions. And from some of those, we get Sanger sequencing with an approach that we refer to as the mini-amplicon approach. We are using like 150 base pair fragments to amplify the control region. Now here you see a Sanger sequence of this first red sample that you saw on the first slide, which had less than 100 mitochondrial genome equivalents per microliter. And we do get a pretty nice control region sequence here. It's not going to the very end of the control region, but it's a good partial control region sequence. Now, with the PEC experiments did early this year, we are confirming this sequence. We are looking at a high number of mitochondrial DNA copies in that size range. We are confirming the sequence, and we are getting additional sequence that it is in agreement with the expected phylogeny. We are looking at a lineage that belongs to haplogroup T1A12, and that is holding 2 signature mutations for that particular haplogroup, and the 15884 is present as well as the 16355, confirming this status. So to me, that is a very nice complementation between confirmation by another method and phylogenetic check. Here is another example. That is a sample that even had more mitochondrial DNA already with the Sanger approach and worked very well. We also use that for the PEC approach, and we are getting the same result, the same control region sequence. We didn't quantify it with the 57 base pair approach, but we saw we get the same control region sequence, plus we get additional information, and again, the phylogenetic check makes it makes a confirmation to that. Well, it's not a big surprise. Now it's getting interesting. Now we're looking at a sample that has 0 quants with 143 base pairs, and we get no result with the Sanger sequencing. So if you put nothing in there, you get nothing out of it, right? But if you put nothing in there with PEC, you get something out of it. Apparently, the nominal values that we get for quantitation is 7 and 2 copies per microliter for the 51 and 57 base pair. Well, that is background. That's the same as the negative controls show us. So apparently, we are looking at a concentration of DNA that is just too low to get a good result. But still, we are getting a useful mitochondrial DNA control region sequence because the fragments that produce this sequence are much smaller. They are between 35 and 45 base pairs. How can we evaluate this result? We can't confirm it by a different technology anymore. Well, we can look at the controls. The controls were all clean. We can try to do a phylogenetic check. It is coinciding with haplogroup H16D if we query that by haplograph, phylotree. And also we can search this sequence in the internet and in GeneBank, and we find a fully overlapping or a full match with an overlapping sequence with 3 sequences in GeneBank, which all correspond to haplogroup H16D. And when I say full match, I mean there is no position that we miss in this sequence with respect to the GeneBank entry, and there is no position in addition to what we get with the GeneBank entry. All right, so as we see when we approach these sorts of experiments, we see that this method is more sensitive because we are detecting fragments that we cannot produce with conventional PCR anymore, but it puts us at a challenge in trying to explain the results and exclude contamination. Well, I would be tempted to say we exclude modern contamination. We were quantifying this extract before and after we did the analysis, and in both cases we did not pick up the 143 base pair fragment. And I would expect to see this if modern contamination was the explanation for this result. We are currently trying to pursue this, and we try to go further down. We are entering a field that is usually worked on by archaeologists and anthropologists, going to very old specimens. These samples are from an Urnfeld culture in Austria. It's on the border between Austria and Hungary. And those remains are about between 1300 and 750 before Christ. If we try to amplify those with if we try to quant those with 143, we usually don't get any signals, but we do get with the very short fragments. And as I speak here, the guys in Innsbruck are currently loading this on the PGM, hopefully, to see if we get a sequence out of this. I would like to change gears real quick and talk for the last 10 minutes about a field that is currently highly discussed in connection with massively parallel sequencing, and that is heteroplasmy. There is this common argument that with massively parallel sequencing, we will be, or we are already in a better shape to detect heteroplasmy at a lower level. We can discuss this in two ways. If that was true, what does this mean for our casework? That's one question which I'm not addressing today. And the second question is, are these new findings confirming or in concert with our experience from Sanger-type sequencing? And for these purposes, it's always very good to have data that have been produced under a high-quality regime to draw conclusions for a new, later technology. So I'm referring to this study with more than 5,000 control region sequences that were analyzed in 2 different laboratories by at least 4 different scientists. This is a long-standing collaboration between the Armed Forces DNA Identification Laboratory in Rockville, now Dover, and our laboratory in Innsbruck. And what we learned from that study is that the control region heteroplasmy rate is about 6% in saliva and blood. These were the 2 tissue types that were used for this kind of study. And we see that we find a maximum of 3 heteroplasmy per individual, not more. If we compare the point heteroplasmic sites to the evolutionary sites in the mitochondrial DNA control region, we see that they correlate to each other. So those that are known to be hotspots, mutational hotspots in human evolution, are also the same sites that show heteroplasmy more frequently than other sites. Very recently, we finished a study where we were doing pretty much the same with full mitochondrial genomes. This is still data based on Sanger method. These are 588 samples that were analyzed for the full mitochondrial genome. And what we find if we look at the heteroplasmy rate, using the same criteria as we did in the study before, we do see an increase 4 times of the heteroplasmy rate in the control region. We do see 24% coding region heteroplasmy rate. Surprisingly, again, a maximum of 3 point heteroplasmy per individual if we look at the entire coding region sequence. And this is in sharp contrast to what we found in the control region, the positions are randomly distributed that show point heteroplasmy in the coding region. They do not coincide with evolutionary hotspots. And that's interesting. In fact, in this study, no single position was hit twice by the same heteroplasmy. It was always a different position. Now let's take this knowledge and look at other data that claim we would be superior in detecting heteroplasmy when we use massively parallel sequencing. And I'm picking one study as an example. It's a study that was looking at the 1,000 Genomes project, and the finding they have is that 90%, not 24, 90% of the individual carry at least one heteroplasmy. They were looking at the 1% minor allele frequency, which means they were detecting and calling heteroplasmy if it was present at 1% or more. While our Sanger study was based on probably a 10 to 20% minor allele frequency, depending on the position in the mitochondrial genome. Sometimes we can go down a little bit further down. Sometimes we are not successful in doing this. More worrying is the fact that they do find a positive correlation between the heteroplasmy rates and the substitution rates. So it's certainly worth to check their data set, which they published in a supplementary material. Now, they do find 15 samples with more than 20 heteroplasmies, and the record holder has 71. 80% of those point heteroplasmic sites occur at haplogroup-specific sites. And now we understand what's going on here. If the mixture is at a signature mutation for a haplogroup, we are very likely not looking at heteroplasmy, but we are looking at a mixture, but a pristine mixture. So here are two samples that we took as an example, the sample 740, where 90% of those 71 point heteroplasmies either correspond to haplogroup L1b or B2b. The first one is a sub-Saharan African haplogroup. The other one is a Native American haplogroup. Or the other sample has 50 and 12 out of 69 point heteroplasmies are diagnostic for either L0A, is again South African, and M7C, which is East Asian. I'm completely making this up, but if you have a student from East Asia working with a sample from South Sahara, you probably would get the same result as we do see here. So if we increase the minor allele frequency to a value that we are used in Sanger-type sequencing, 15%, then we are losing all the point heteroplasmies that they are reporting. And the same is true for 25% for the other sample. That tells us, with this little method, we can even predict the contamination rate in those samples or in those results that were presented by the 1,000 Genome Project. There are a couple of other studies. So what we tried to do, and we just submitted this to a review article in Trends in, well, I think it's just in an FSI Genetics special issue. We were looking at all the published data sets so far that had the detection of heteroplasmy in mind, and I'm circling our study, which I'm using as a reference here, with a 24% point heteroplasmy rate. The green dot that you see on the top level shows the percentage of coding region point heteroplasmies observed only once, which in our case was the case for all heteroplasmies. And the black diamond that you see at the bottom of this graph tells you about the maximum number of point heteroplasmies observed in an individual. And those two measures are very informative if we look at different studies. So for example, there is another study here by Bruce Badoli's group. Jonathan King has produced this. It's very similar to the study that we found, but this is using next-generation sequencing as a technology, while this is using Sanger sequencing. If we look at the study from Ramos, completely different samples, completely different technology, but come to very similar conclusions. Also, Lee from Mark Stoneking's group are arriving at similar data. If we look at the SOSA study here, we do see a much higher level of point heteroplasmy using the 10 to 20% minor allele frequency. However, we also see multiple hits in a single individual. In that case, some of these positions, we think, are artifacts of the allele calling software, of the alignment software that was used. Now, if we look at these data here from Konieczna, I hope I pronounced that fairly correct, a group from Poland. They also fall in the same ballpark, using a 2% minor allele frequency to detect point heteroplasmy. Now, the real problematic ones are these ones, the DeRoma study, because that is far too high. The green dot is far too low. There are too many point heteroplasmies observed multiple times, and some of them are observed very often. And the maximum number of PHBs in a single individual tells us that they were very likely using contaminated samples. Now, here is a study that the group, Mitchen's group in Penn State did. They were looking at a 1% minor allele frequency. If we use the same data set and put this minor allele frequency at 20%, we find results that are in line with all the other studies, and we get additional sensitivity, apparently, if we move up to 1% and even below 1%. And there is this study. It's a study that is already in the revised form. That's the study that I presented as an example before. Even the revised data do not correspond with what we would expect if we were using, or if we were compare it to current Sanger practice. All right, that's the end of my presentation. A quick summary here. We do see that the PEC is producing concordant results to sequences where we compare. It's good for shorter DNA fragments, so it's a very nice feature. It seems to be more sensitive. However, it puts us at a challenge to explain the authenticity of the result. So we need to find different argumentation that is probably used in the archaeological field and settled there, but that is probably new to the forensic field. And in terms of point heteroplasmies, we appreciate these kind of studies, and we are continuing these kind of studies, but we should be cautious with being too optimistic here. A phylogenetic check is certainly worth a control on point heteroplasmies, and the random distribution, the coding region, is also something that we would expect when we look at such kind of data. I'd like to thank funding bodies, particularly the Eurofortune group and the NIJ group who have funded this work. I'd like to thank my co-workers, again, Myra and Katrina. Now they are laughing again, as you see in the slide, doing fantastic work. Petra is working on the old bones. Tina is working on the PGM. Harry is practically working on everything in our laboratory. And finally, I would like to thank ABI for inviting me here and for you for listening. Thank you very much.
Are there any questions? Are there any questions from the audience?
Hello. So I'm wondering, do you have to make an hypothesis about the molecular mechanism that's at the origin of heteroplasmy? I guess, I don't know if there is a consensus, I guess that might change the interpretation of whether your mutation is a hotspot, evolutionary mutation, or can be a mixture, but kind of an embryonic mixture. You know what I'm talking about?
Yeah, well, the mechanisms for heteroplasmy are relatively poorly understood. We know that those mitochondria go through a bottleneck, so also mitochondrial DNA is going through a bottleneck. And during this bottleneck phase, spontaneous mutations can be increased in their amount after they pass this bottleneck. And this is how we explain, for example, that in different human tissues, we do different segregation of this heteroplasmy type. As far as and what I probably should add is, what we know from medical genetics are those deleterious mutations that are present in heteroplasmic form. They never reach a high penetrance in the population because there is this self-correcting effect during the oogenesis and during this bottleneck effect to cut down this population size to this low level. If we consider a point heteroplasmy or heteroplasmy in the forensic context or in the analytical context, then we need to be cautious in discerning actual signal from a real mixture from two samples, from an artificial signal that may come from something that slipped through the alignment process, polymerase errors, other artifacts that may arise during the synthesis of the results. And I guess, but that's my personal view, I would not be in favor in trying to reach down with heteroplasmy too much in terms of mitochondrial DNA typing, because it's getting extremely difficult to get a handle on it. And if we look at the outcome, what can we do with this? At least so far I didn't understand what's the consequence, because we are ignoring point heteroplasmy and length heteroplasmy largely when it comes to the interpretation of evidence. We are not using this as an exclusionary marker.